Biology Part 206. A condensation continues, the nuclei shrink, finally disappear. Although the nuclear organizers, that is the chromosome regions responsible for nucleolus formation, may remain visible throughout the deviant. Prophase is also marked by the development of an arrangement of microtubules known as the mitotic spindle. At the beginning of the prophase, the cytoplasmic microtubules, which form the cytoskeleton described here in our tutorial, are broken down, allowing the cell to round up into a spherical shape. This breakdown provides a large part of the proof of tubular molecules, which are gradually incorporated into new microtubules. The first of these to appear are called epolar fibers, and they radiate from the pair of mitotic centers which move slowly towards opposite poles of the cell. In animal cells, a structure called an aster is formed at each end of the spindle, with the fibers radiating in all directions. A centriole pair is located inside each mitotic center. The exact function of the centrioles is in some doubt, as in experimental cultures, deviant continues normally even when they are destroyed using a laser microbeam. In plant cells, the mitotic centers are less well defined, and no aster or no centrioles are completely absent. Thus, the centrioles are not directly essential for the spindle formation. The prometaphase begins with the sudden distinguish of the nuclear envelope. The spindle moves to occupy the central region of the cell, and the centromere of each chromosome forms the kinetocores. These structures lie on either side of the chromosome so that the kinetocores, microtubules and kinetocores fibers project in opposite direction. The fibers interact with the spindle, often resulting in agitated chromosome movement. Metaphase. All the chromosomes are arranged with their centromeres near the equator of the cell. Place of alignment is called the metaphase plate. The kinetochore microtubules lie parallel to the spindle and pull towards the opposite poles, holding the chromosomes in place under tension. Compared with the other stages, metaphase may last for a long time. Anaphase. Anaphase begins when the kinetochores at each centromere suddenly split. The kinetochore fibers now slide towards the opposite poles of the spindle, actively pulling the chromatids apart. At the same time, the spindle lengthens. The biochemical basis for these movements is not fully understood, and the net effect is to form two daughter nuclei, each genetically identical to the other. Not that both of these contain the chromatid from each parent chromosome. The two nuclei are identical because the chromatids themselves are identical molecules of DNA produced by replication.